viruses in general, but particularly RNA viruses like HIV and like Ebola, do not have proofreading activity in their polymerases. So they mutate very, very quickly. That gives them an enormous selective advantage against us. And it also gives them a huge advantage to be able to essentially become resistant to the drugs that we throw at them. So, and we'll talk more about this later, but that is absolutely why you need to throw a multi-drug cocktail at HIV. AZT that we talked about yesterday, right, targets the polymerase, targets the reverse transcriptase. Reverse transcriptase mutates quite quickly to avoid AZT. So you have to keep pressure on retroviruses, particularly from many angles, all at the same time. You target the polymerase, you target the protease that cleaves, you target the integrase, you target many, many different enzymes that it's utilizing all the time. And by doing that and keeping selective pressure on multiple points, you keep it at bay. That's basically what you have to do. You have to keep doing that, right? So that's the other thing that you're going to learn lots more about in micro. But once you're on a cocktail of inhibitors, you basically got to stay on it. And you got to have a high level in your system, or else the virus will start coming back out even stronger. So essentially, you've got to keep the virus at very, very low levels, essentially undetectable in your system, in your bloodstream, so that it's not spreading around again in your body. Now we know, and unfortunately, I'm not going to do the retrovirus lectures this year, I don't think, but there are a lot of therapies that we're starting to look at to try and see whether or not we can flush the virus out of latency. And that's something that we'll talk a little bit more about later on when we're talking about the gene therapy stuff. When we kind of touched on it with this CCR5 but we'll talk more about that. We'll talk about it later. Okay, I wanted to talk about one more little thing about replication as a whole before we talk about telomeres and talk about reverse transcription. So this guy, the helicase, is opening up the strands, right? Plowing through those strands. And think about this, right? So we know from our conversation this morning that we've got right-handed helical turns in front of this helicase, right? It's not that it's just plowing open two strands of DNA that are flat on the surface and all we have to do is peel them open, right? That's not the case. It's helically wound. And if anybody, who here has ever seen a telephone cord? Right? It's not, that. Maybe you haven't used it, but you've probably seen one, right? And you can come into my office and you can see how the damn thing coils on itself all the time, right? So it's got coils, but as soon as you put more pressure on those coils, and especially with the helicase, with opening up those strands, you're getting a lot of pressure built up on the normal helix and on the turns of the normal helix. Well, as soon as you create enough pressure on that, it's going to start going in the opposite direction, right? So those helical turns are all of a sudden going to create turns on themselves in the opposite direction. Those are called supercoils, okay? And they're called positive supercoils because they go in the opposite direction of the coiling helix. Okay, so we're introducing positive supercoils into the DNA that's in front of the fork by pulling the fork open, okay? Now, very, very quickly, this thing is going to get stalled out by all of those positive supercoils that are ahead of it. Okay? So we've got enzymes. We got an enzyme for everything. And this enzyme, these enzymes are called topoisomerases. Right? So think about the topo map, topographical map, right? Shows you all the peaks and valleys. Well, that's what this is. It's going after those big old honking mountains that are in front of the replication fork. Okay? So the topos essentially come in, and they're there to remove those positive supercoils. Okay, we've got two types of topos, topo 1s and topo 2s. So topo 1s will cleave one strand of the helix. Okay, so think about this. You got a coil, right? You cleave one strand, and you basically just start unwinding it in the opposite direction to get rid of the supercoils. That's what a topo 1 will do. Topo 2s, which are much, much more common, will actually cleave the entire helix. They'll cleave both strands, and think of this as a positive supercoil. If we cleave the strand, or cleave it here, we can actually pass the coil through that cleaved piece of DNA, and in essence, remove that supercoil by passing it through the other strand. It's kind of weird to think about visually, but so we've got our two strands, and if we make a gap in between them by introducing a double strand break, we can bring past the actual other strand through it, the double-stranded other strand through it, right? Does that make sense? A supercoil is a double-stranded DNA on top of another double-stranded DNA. If we open up one of those double strands, we can pass the other double-stranded coil through it and unwind. In other words, we're just getting rid of that excess winding, those positive supercoils. So topoisomerases are incredibly important enzymes. As you might imagine, there's something that we can target. Okay? So turns out that bacterial cells have a very specific topoisomerase. It's called gyrase. And we have we have drugs called what? Called quinolones, thank you, that actually target one of the subunits of gyrase. It's very, very specific for gyrase. It does not work on our topoisomerases. So it's specific for one of the subunits of gyrase itself. It actually keeps it competitively binds to gyrase after it's actually made the cleavage. And so it binds to it, and then it won't let the strands go back together. So it's actually really super effective for inhibiting DNA replication in bacteria. Sir, so gyrase is the topoisomerase in bacteria, and the quinolones, particularly ciprofloxacin, which you've probably heard of, Cipro, targets very specifically the gyrase, one of the subunits of gyrase. So that after it's cleaved, the two strands, starts passing it through, but it doesn't allow the two strands to come back together. So in essence, it makes a double strand break that can't be reannealed. So it's really super effective against replication. So it essentially shuts the cell down. So it's really good, and it just targets them and not us. Yay, it's another us versus them happy story. Okay. As opposed to us versus them sad story. That's where we're at with the bullet. Us versus them sad story. Okay. I want to have another piece of candy, but I don't have any. So instead, we'll keep talking about this. Preventing re-replication. All right. So we're doing this replication of our DNA, right? We have to copy all of it. We know that in one round of the cell cycle. Okay. So right? Remember the cell cycle. Yes? We know the cell cycle? Do we know cell cycle stuff? A little? In general. Should we review a little, maybe? Well, we'll review later, but we can do it. How do we? I need to skip out of that for a minute. This. Okay. Someone was writing on the board. Can I erase this? Yeah. Okay. Don't pay any attention to that. We're just going to do a little cell cycle review. Okay. So this is G1, and this is S, and this is G2, and that's M. Okay, so that's a little bit distorted. This is a little bit longer than that. Okay, so M, mitosis, right? And S phase is for replicating. And then we've got these phases called G1 and G2, which are resting phases, if you will. Resting and evaluating. And we'll talk much, much more about this later, so I'm not going to belabor that point. But in essence, what happens in S phase? Well, in S phase, we've got to do all of the replication that we just talked about. Okay? Once that's done, the G2 phase is our, like we said, evaluating fidelity, completeness, broken strands, damage, anything else. So this is in essence saying, okay, we replicated it all, looks like a good copy, everything's intact, we've got a good set of chromosomes, now we can move it through into mitosis and divide. Once. This happens one time during each replication cycle. That's essential for us to be able to control that and make sure there's not re-replication going on, right? We don't want to fire those origins, all those hundreds of origins that we have across our chromosomes, more than once in a cycle. Okay? So there's a lovely way. That makes sense, everybody? There's a lovely way. Why do I keep walking over there? Because in the other room, it's over there. In our cells, there is a way to do this. And here's the bottom line. There's a bunch of proteins that are involved in regulating this. Once the origin is fired, they are shipped out and taken care of so that they cannot rebind to that origin until the next round of replication is set. Okay? So let's look at how that works. There are a lot of players on here. Don't worry about the specific names of these proteins. I'm just going to throw them out because as I'm pointing at them. But don't worry about all the players and the names. Just kind of get at conceptually what's going on here. Okay, so we have our origin, right? We know we have a bunch of them, and we know we have to recognize them because that's where we're going to open up the DNA, right? So there's a complex of proteins. There's 6 proteins that are called origin and replication complex proteins. There's 6 of them. 2 through 6 bind all the time to the origins. They're always there. ORC1 is the guy, the swing guy. He has to come in to start the replication process going, to fire that origin. So we said it's kind of a crazy term, but that's actually what you say. You're firing that origin. You're essentially, you know, like a little firecracker. You're setting it off so that it goes in both directions, okay? So ORC1 has to be in the complex in order for us to be able to fire. That's step 1 in essence. And that happens during G1 as we're setting the stage. Several other proteins have to come in and bind to that site. The MCMs are proteins that are going to help to open up the DNA and to start the replication process going. And these two proteins here, CDC6 and CDT1, again, you don't need to remember the names. They're called licensing factors, right? So think about this, right? The guy who gives you your driver's license, who you have to go and take the test, right? He's your licensing guy. He's the guy who says, yeah, you can do this. Good to go. You didn't hit any cars while you were driving around. He gives you your license. That's what these guys are. They're allowing the MCMs to load onto the replication origin. Okay, they're actually helping them to get on. Once everybody's in place, one of the S-phase kinases comes in and phosphorylates several of the MCM proteins. Okay, so here, and this is something that we are going to see lots and lots of times, phosphorylation of a protein oftentimes activates it. And this is exactly what's going on here. We're activating the MCM complex so that we start firing the origin. Right, so these are all guys, and this is kind of conceptually, right? It's like, how many clowns can you get into that tiny little car, right? There's a lot of proteins that are sitting around on this origin, and then the whole replication machinery's got to get in there too. So there's a lot going on at this origin of replication. Just thought about that. Okay, so here we are. We're firing, right? So we're set, we're ready, everything's good. We're firing the origin. We start replicating, and the origin is moving out. The forks are moving out. Now, we want to prevent anybody from sitting back down on that origin and firing it again. End of story. That's what we want to prevent. So how can you do that? Well, first off, degrade ORC1. There's a really good way to do it. Because without ORC1, you cannot start this process. So that's what we do. We degrade ORC1. We also degrade CDC6. We sequester CDT1 with a protein called geminin. And we also sequester the rest of the MCM complex. So again, you don't need to remember who gets bound by what or anything like that, but just remember that there's essentially degradation going on, there's sequestration going on, so that we don't have all the players anymore once we're past firing those origins. Now obviously this is a sequential process. There are early firing origins and late firing origins. But in essence, once we're done with that firing, we degrade all this stuff, we sequester all this stuff, so that then the only time that we can start this process again is after we've moved through mitosis and into the next G1, where ORC1 is synthesized, geminin is degraded as we pass through mitosis so that CDT1 is available again, CDC6 is resynthesized, the MCMs are let go and brought back into the picture. Okay, so again, don't need to remember who all the players are, where they are, but just that the process is essentially everybody's there, party started, cops show up, everybody has to leave, and here we go again, right? They're not until the cops are gone can everybody come back into the pool. Okay? Make sense? Okay. Really super important. And a lot of these players are mucked around with in cancer, particularly the kinases, the proteins that are involved in actually firing the origins are present more often. particularly ones that target them and not us, right? So we just figured it's just a recap of who we've talked about so far, right? So photomides, remember, are the guys that target PABA. Our quinolones we just talked about, inhibiting gyrase. Our acyclovir and ganciclovir and AZT are our inhibitors of polymerization of the viruses, right? There are nucleoside analogs or chain terminators. And our anti-cancer drugs, methotrexate, we didn't talk about these, but I'm throwing them out there right now. We actually have drugs in our anti-cancer arsenal that are topo 1 and topo 2 inhibitors, okay, for our topo 1s and topo 2s. So these guys, again, have to be used sparingly. All these drugs are super toxic, right? We know that this is going to nuke our cells, but it's going to nuke the rapidly dividing cells much, much more frequently. So again, right, if you're inhibiting the topoisomerases, you're going to be inhibiting DNA replication. So in a fast, rapidly dividing cell like a cancer cell, that's going to be much, much more effective than a cell that, every 24 hours or so, maybe 36, we replicate, right? Those cells are not going to be hit nearly as hard. Okay. Bing, bang, boom, 20 minutes. Yeah, I can get through this. I can do that for 20 minutes. All right, let's talk about telomerase a little bit more. This is a really cool enzyme, and it's really essential because of the following. Right now, think about the ends of our DNA. Right? So this would be so easy if we had circular DNA. We could just replicate around in a circle all day long, rolling circle just like viruses, and it's like good to go. You just string out concatenators and we're happy. Everybody's happy. We don't do that, right? We got ends. And at those ends, think about this, right? You've got ends, and if you've done your homework right, at the end, there should be, right, a primer. Because we have to have an RNA primer every time we synthesize a piece of DNA. So there's going to be a primer at that end, but it's got to be removed, right? Because we can't have RNA in our DNA sequence. So there's always going to be a small region at the end, at the 5' end of both strands, at the ends of the chromosomes, that is not there, that is removed, because you had to get rid of the RNA primer. So if this happens, every time you replicate, you're losing sequence, and sooner or later, you're going to butt in to where there's actually a gene encoded. And that's going to be super problematic for you. Okay? So, and it's actually not just the primer that's removed every time. So it turns out about 100 base pairs are removed from the ends of our chromosomes every single time we replicate. That's because of some extra exonuclease activities we don't need to go into. Okay, so 100 base pairs every time we replicate. That's a lot. And our cells replicate fairly regularly. Okay, and so pretty soon you're going to be running into sequence that you don't want to be losing. So, smart guys that they are, that designed us, and girls, decided that we need to have some protective sequence on the ends of our chromosomes. Why not just shove a bunch of non-coding sequence onto the ends, little repeats that we call telomeric repeats, that we have 2,000 copies of, so that every single time we do this, okay, yeah, we lose a couple of those, but it's not really that big a deal. And sooner or later, we are still going to end up getting close to coding sequence. The cell knows that at that point, it should stop replicating. And that's a process that's called senescence, right? You probably have heard of that term. So, senescence doesn't mean death. It just means you're getting old, right? So, right, little Grammy and Grandpas, they're not just out running all around anymore, you know, playing ultimate frisbee with you and getting concussions. They're probably hanging out on the porch, sipping iced tea or cocktails, depending on what time of day, and just relaxing, right? And that's what a senescent cell does. Still makes proteins, still functions just properly, it's just not dividing anymore. I'd like to think of myself in retirement as senescent and not just kind of hanging, doing the things that I like to do. Not having to divide, get up and give a lecture. How do we get that, right? How do we get to a point where we've got this sequence on the ends of our chromosomes that's going to protect it? Well, it's through the process of telomerase, okay? So, think of this, okay? At that, dang it, I have to climb on the bench again. Because we can't see it. All right. Here's our 3' end, right? Now, if you look at what that 3' end looks like, it's down here. And you can see that it's encoded by telomeric repeats, okay? So, forgive me, this is actually, I think, the Saccharomyces cerevisiae repeat. Our repeat is TTAGGG. That's actually in your notes there, so don't worry about it. But same exact principle goes on. Okay, so our telomeric repeats are synthesized on the end of this 3' strand here. Okay, so right, 3' means it's got a hydroxyl there, right? So this is actually acting as the primer to start our telomeric synthesis. So, what happens? Telomerase is the enzyme that's going to add these telomeric repeats, okay? So it turns out telomerase is actually super cool, super quiet reverse transcriptase, okay? Because it actually utilizes a little strand of RNA as a template for the synthesis of the telomeric repeat, right? Because think about this. It's adding these strings of telomeres adjacent and opposite to nothing, to space, to air, right? So it doesn't have a template of DNA that it can be copied. So it has to provide its own little template inside its catalytic surface to act as the template for this telomeric repeat to be synthesized, okay? It's actually a piece of RNA that's encoded in our genome. That's a specific subunit of this, what we call riboprotein, right? Because it's a ribonucleotide protein. It actually has an RNA component and a protein component. Okay, so telomerase is actually a reverse transcriptase. It's taking an RNA template and synthesizing a strand of DNA. So how's that work? Well, here's our little template. It's copying right from that 3' hydroxyl, it's synthesizing the telomeric repeat. Well, it's only 6 nucleotides long, for heaven's sake. That's not going to get us very far when we're thinking about chewing up 100 base pairs every time we replicate, right? So we got to keep doing this over and over and over and over again. So we take this, okay, we've synthesized it, we ratchet over the space of 6 nucleotides. So now the telomerase ratchets over and it recopies this, putting on the TTAGGG, so putting on the last G next to this synthesis. So does that make sense? So we got a copy, we ratchet, we copy again, we ratchet, we copy again, we ratchet, we copy again. This is really cool perspective. Right? Does that make sense? We're just going 6 nucleotides, 6 nucleotides, 6 nucleotides. It's just like when we're reading an mRNA transcript to make proteins. You got to ratchet the codon distance, right? Anticodon distance. So it's the same kind of ratcheting thing, and we're just making hundreds of copies of the telomeric repeat. So that then, when we replicate the next time, and we have to take off several nucleotides on our 5' end, well, we still have plenty there. So we're chewing these back every time we synthesize, but we've got lots and lots to spare. But again, over time, we do get close enough to a point where we're going to get close to coding sequence, and the cell knows that, it senses that. There are enzymes there to sense the length of the telomeric repeat. Shuts it down so that we're not replicating it. Okay. So, crazy as this might seem, right? Telomerase, super active during development, right? So as our embryo is being formed, as we're forming all the new organs inside that embryo, telomerase is working. We got nice, long telomeric repeats. But then it shuts down. Okay, so telomerase is not active in the large majority of our cells. Okay, it only remains active in rapidly dividing cells, like stem cells, right? Hematopoietic stem cells, hair follicles, crypts, all the guys that get hit when we're treating people with anti-cancer drugs, right? Intestinal crypts, things that are rapidly dividing and always rapidly dividing. Those are the cells where telomerase is still present and working. Most other cells, any somatic cell is shut down with telomerase. So, here's the kicker. Many cancers, many tumors, we actually see telomerase turned back on. Okay? So and that makes sense because that cell is rapidly dividing. It needs to actually synthesize telomeres to get past that senescence phenotype. So it's got to make these big old honking telomeres, and then it can replicate like crazy. And you actually can see that very easily. There's gels that you can run that you can see the telomeric repeats, and you can see it expand like crazy when you run the tumor DNA tissue. So how does that happen, you might ask? Well, it turns out that it looks like in many melanomas anyway, large majority of melanomas actually, and in some, about 10 to 15% of other types of cancers, you actually get a mutation in the promoter sequence of the telomerase that now allows a particular transcription factor called X to sit down and drive the transcription of telomerase, which normally would not do that. So promoter mutations can be deadly for this. But the other cool part is, right, well, if we know that telomerase is getting turned back on in these cells, couldn't we potentially target the telomerase and shut it off? That's exactly what we're trying to do. And there are drugs in trials now, telomerase inhibitor drugs that are designed to essentially target the rapid proliferation of the tumors, and you target them right into the tumors. Nicholas.
So you said that for telomerase cells, it's a mutation in the promoter sequence that just causes that.
Allows the telomerase to get turned back on. Yep.
And then you also said telomerase was a ribonucleotide
No. It's a ribonuclear protein, right? Because it's got a ribo component and a protein component. Make sense? Yeah.
I've read articles about people discussing like anti-aging things where you add.
Wouldn't that be lovely?
Are they trying to target telomerase so that it still works, or?
No, they're trying essentially to target the sirtuins, which are proteins that control the telomeric repeats and repair at the repeats. So it's not trying to turn telomerase back on. Nope. That would be scary. Okay. So we have a really kind of cool phenomenon that turns out from these telomeric repeats that I want to just make you aware of, right? So be a little overhang at the ends, right? So we're always still going to have to remove that primer off of the 5' end. So there's always going to be a little bit on the 3' end of the opposite strand that's going to be single-stranded, always. And that, my friends, is prime candidate for exonuclease attack, right? So there are lots of enzymes in our cells that don't like these kind of things floating around. And they could potentially attack the end of our chromosome and start chewing it back. And that's not something we want to happen. And so we've really got to protect this telomeric sequence to keep it from getting attacked by our own bodies, okay? And so there are a complex of proteins, and again, you don't need to remember the names of these guys, called shelterin, right? Isn't that a lovely name? Shelterin, and it's there to shelter or protect the telomeric ends. And so it works by a complex of proteins binds to both the double-stranded helix, the double-stranded part, and then there's a protein called PART2, which binds to the single-stranded DNA. And it, in essence, loops that guy around and back in so that the single-stranded part actually invades the double-stranded helix in the telomeric repeat region, so that we actually have a triple helical structure there, which is really kind of a cool thing. It's not a phenomenon you see very often, but this has got 3 strands of DNA in this little, what we call D loop. Okay, so the whole thing is called a T loop. So the T loop structure is very, very uncommon. This is something that is just seen in, essentially guys that have chromosomes like this, like us, and all vertebrates have these guys. And the D loop is really pretty phenomenal in keeping and sheltering that single-stranded DNA so that it's not subject to cleavage by our exonucleases. So this is a protective measure. Obviously, all this crap's got to get taken off when we replicate the DNA, right? So all this stuff has to get removed, and that's signaled for as we're starting the replication process. These proteins are removed, the thing unwinds, so that we can actually replicate all of them. But our telomeres are protected like gold. They're really very, very highly regulated and very, very closely watched because, right, think about this, and we'll talk much more about this when we talk about repair, but double-stranded ends, right, they like to go together. They can be fused relatively easily, and we have enzymes and we have DNA repair machinery that's looking for those kind of things, right, breaks that happen in the DNA. We've got what we call double-strand break repair machinery that's looking for ends to seal them back together so that we can go on and replicate and go on and transcribe through that DNA. Well, these chromosomal ends would look just like the template for a double-strand break annealing if they were not protected by the sheltering complex and by several other proteins, which we're not going to go into at the moment, that bind and protect these ends. Okay, so we keep them like little babies, swaddled up so that nobody, big bad exonucleases or anybody else can attack them. Let's just talk about this for a minute. This is a disease, dyskeratosis congenita. It's a crazy sounding disease, but it's kind of bad because what happens is in dyskeratosis congenita, the people that have this disease have a mutation in the gene that encodes the little piece of RNA that's used as a template for telomerase, okay, the RNA subunit. And that gene, when it's mutated, you can't make the RNA, or at least it has a mutation in it, so that you essentially have a non-functional telomerase. when you have a non-functional telomerase, that can be bad, right? So you have very, very short telomeres, and things that need to rapidly replicate, like bone marrow, and your skin cells, your underlying skin cells that replicate quickly, the nail beds, your hair follicles, all the things, again, that are targeted when we hit with anti-cancer drugs, all those rapidly dividing stem cells are going to be very, very few in these kids that have dyskeratosis congenital. So, as you might imagine, it's not a disease that can be survived for a very long time. Okay? So very, very problematic. But, right, this is something we could target with gene therapy too. Just a single mutation in a single gene. Okay, ooh, 5 minutes. Yay. All right, we're going to start this. All right. Telomerase is a reverse transcriptase. It's in our bodies. It actually has a tiny little bit of proofreading activity, just so you know. This reverse transcriptase that's encoded by retroviruses does not. Absolutely does not. Okay? So I just wanted to give this to you as a little bit of a introduction to reverse transcriptase and to retrovirology, because you'll see this much, much more when you do microbiology. But it just gives you a glimpse into how ridiculously complex retrovirus life cycles are. Okay? So first off, you're coming in as a retrovirus with 2 copies of a single-stranded RNA. Okay? Plus stranded RNA. Who knows what a plus strand of RNA is? No idea. Right. Okay. Divergent number 1. Or number 2. Let's talk about plus stranded RNA for a minute, because it's something that you're going to absolutely need to know. All right. Mute this for a minute. Oh, crap. No. Just mute it. All right. Okay. In the dogma of virology, there are multiple different genomes that we have to worry about. And this whole concept of plus stranded RNA kind of comes in through the virology life cycle. Okay? So, think about an mRNA. Okay? So an mRNA is considered a plus strand. Okay? So think about what this is coming off of, right? So in a double-stranded DNA molecule, you have 2 strands, right? They're opposite polarity, opposite sense, complementary, whatever the heck you want to call it. But in terms of this, we're talking about opposite polarity. Okay? One is a plus strand, one is a minus strand, right? Because plus and minus are opposites. So if you use the minus strand as a template, and we're just using that for a second as an example, and you synthesize your mRNA, right, 5' to 3', what is that going to be? Going to be plus, right? So that is the polarity that you have to think about. When you think about mRNA, it's a plus-stranded RNA, okay? Now, let's think about that in terms of a virus for a second, okay? A plus-stranded RNA virus, single-stranded RNA, is going to be So David Baltimore came up with this polarity scheme, and I think it actually makes an enormous amount of sense. Okay, so a plus-stranded RNA is the same polarity as an mRNA. Now think about this in terms of a virus. A virus is coming in and has to utilize all of the cellular machinery to make itself go, right? It's got to use particularly it's got to use the translational machinery. So if its genome is coming in and looks just like an mRNA, bam, it gets deposited into the cytoplasm and immediately gets translated. That is a bonus if you are an RNA molecule, if you are a virus. Immediate translation means you don't have to carry as much with you. It means you can translate your proteins quickly and efficiently when you come in. Okay, so that's plus-stranded RNA. Now we got a little bit of a upheaval when we talk about retroviruses. Okay, so single-stranded RNA viruses, this is what happens immediately. First step, you get translated. That's not the case with a retrovirus, right? Yes has this polarity, but it is first going to get retrotranscribed to a double-stranded DNA, okay? So what does that mean? Now we're going back to this land here, right? If I have my transcript, my single-stranded RNA, that's plus strand. This is my retrovirus. My single-stranded RNA, now instead of getting translated in the cytoplasm like it could be, if it was a normal just RNA virus, what's going to happen? Reverse transcriptase, oh, that's blue. Reverse transcriptase is going to come along and make the first DNA strand. Which is minus in polarity, right? Because it's copying the plus strand. And then it's going to synthesize a second DNA strand. That is plus polarity, so it's the same sequence as the RNA that came in. We'll walk through that whole process next Monday. We'll walk through the whole process of how that conversion happens. But I just want to get you thinking in terms of plus and minus and how does this work. And if you're a virus, you got two options, right? If you're just a regular old RNA virus, you're shooting just for translation. But if you're a retrovirus, you actually want to look like a double-stranded DNA so you can insert into the genome and stay there forever. Okay, so different thought processes for different viruses. Think about that over the weekend, and we'll talk about it next week.